mutation. So what is mutation? It is a sudden heritable change. So sudden, it will happen at any time, right? So it is a sudden change. And heritable change, what do you mean by heritable change? That it can be transmitted. So once the change occurs in the DNA, that is further transmitted to the offspring. So it is a sudden heritable change occurring in DNA sequence. So if there is a particular DNA sequence and if there occurs a change in the sequence in the chromosome, the change will be transmitted to the offspring. So that is the mutation, right? It is a sudden heritable change occurring in DNA sequence that results in changes in the genotype and the phenotype of an organism. So as a result of the change in the DNA sequence, there will be a genotypic change as well as the physical expression of the phenotype. That is the phenotype of an organism. Okay, so that is the mutation. So recombination and mutation, it leads to variation in DNA. So it leads to, as a result of recombination, there will be formation of the new genes, that is new combination of genes, and mutation, it leads to change in DNA. So all these leads to variation, right? So the variation in the characteristics that occur due to recombination and mutation. So there are different types of mutation like point mutation and frame shift mutation. So what do you mean by point mutation? That is, the mutation occurs at a particular point, right? Only in one base pair. So it is a mutation due to change in a single base pair of DNA. So for example, this is the original sequence. So all the other bases, that is TAA, you know all these, right? Thymine, adenine. So these are all the nucleic acids that are present, right? Here the thymine is a base which is present here, and adenine, cytosine, and then thymine. So here what occurs in the original sequence, here this T is changed to C, right? So here thymine is changed to cytosine. So it is a change in a single base. Actually, there occurs a change only in a single base. All the other bases are the same as it is. So the point mutation is due to a change in a particular point alone. So it is due to a change in a single base pair of DNA. Example is sickle cell anemia. So because of a single base pair change, there occurs a change in the particular amino acid, right? In the case of sickle cell anemia, glutamic acid 2 valent. So there occurs a change in the amino acid. So as a result of the change in amino acid, and that leads to the defective protein synthesis, right? So that leads to a disease which is called the sickle cell anemia. So this is a point mutation as a result of a single base pair change. And what do you mean by frame shift mutation? That it is the deletion or insertion of base pairs resulting in the shifting of DNA. For example, so these are the original nucleotides that are present, right? That is A, C, G. Know the 3 nucleotides, that is, they form a genetic code, and it codes for the amino acid, that is threonine arginine. So what happens as a result of deletion or insertion of a particular base? So if a particular base pair is deleted, so it leads to the defect in the reading frame. So what do you mean by the reading frame? That is, if A is deleted here, now the next 3 bases will group together. So it will become CCA, and instead of coding for the amino acid threonine, it will code for arginine. So the entire reading frame will be affected as a result of the deletion or the insertion of base pairs. So it results in the shifting of DNA. So beyond the point of the deletion, what will happen? The amino acids will change as a result of the protein that is produced will also change, right? So loss or the gain of DNA segment causes chromosomal abnormalities or aberrations. That is, there occurs a change in the chromosome structure and the number because of this loss or the gain of DNA segment. So these chromosomal aberrations are seen in cancer cells. So these chromosomal abnormalities we can see in the cancer cells. So as a result of a loss or a gain of a particular segment. So the chromosomal mutation may be either due to loss of the segment, or it may be due to the gain or the duplication, or there may be in some cases it may be the translocation. So translocation is the mutual exchange of the chromosomal segments between the non-homologous chromosomes. So between the chromosomes, there may be an exchange of genetic material or the chromosomal segments, right? Or it may be due to inversion. So if it is an inversion, the chromosome breaks at 2 points. So there will be nucleotides and it may break at 2 points. And then this broken piece, it rotates through 180 degree and then it reunites in a reverse order. So this nucleotide will come over here. So it breaks at 2 points and then it rotates through 180 degree and then they will reunite in a reverse order. So there occurs a change of the bases which are present here in this position, right? So that is one type of a chromosomal mutation. So it may be either due to addition or deletion or translocation or inversion. Okay.